My name is Ben Loveday. I'm a marine earth observation scientist and oceanographer working for Innerflare in Germany. Today I'm going to give you an overview of a Jupyter Notebook tutorial on the use of machine learning for water type classification, in this case using a Naive Bayesian classifier. This notebook was written by myself, Dr. Hayley Evers-King from EUMETSAT and Dr. Lauren Bierman from Plymouth Marine Laboratory. Let's take a look. At the top of the notebook, the first thing you'll find is a short video, the one I'm recording right now, which will give you an overview of how this notebook is going to run. You can always refer back to this at later times. So let us begin. First and foremost, why are we doing this? Why do we care about coastal ocean waters and optical complexity? Coastal oceans are where our society meets the sea. They're where we tend to engage with our ocean both recreationally, through swimming, sailing, diving, and also where we tend to engage with our oceans economically through aquaculture, fisheries, ports and shipping. However, they're also the most complicated parts of our oceans in some ways. They have lots of different components in them. They have algal populations, they have sediments, they have coloured dissolved organic matter, or as we refer to it as CDOM, all these different layers of complexity that can make with extracting signals from our coastal oceans very, very difficult from satellite. This workbook will show you how we can use machine learning approaches, in this case a Naive Bayesian classifier, applied to Sentinel-3 AltG data to address some of this complexity. The machine learning algorithm we're going to use is from the scikit-learn toolbox in Python, and it's the Naive Bayesian classifier. It's a relatively simple method that's based on Bayes' theorem, and we're going to use it because it's fast and it's simple and it requires relatively small amounts of training data. There are key assumptions here. The main one being that the features it classifies are independent, and we will come to this later. We're going to apply this algorithm to data from Sentinel-3, and we're going to use two input products from Sentinel-3 and one input of a radiative transfer model for our training. We're going to use an AltG full resolution level 1 scene for the North Sea for the 16th of April from 2021. We're going to use an AltG full resolution level 2 scene for the same time period, exactly the same coverage. And we're going to use this radiative transfer model spectral output to simulate remote sensing reflectance, and this is adapted from work done by Evers-King et al. In 2014. If you want more information on the AltG products or the training data that we're using, you should refer to the further resources section. So to give you an outline of our notebook, what are we going to cover? We will start with an introduction to AltG data. We'll follow this with an introduction to how we interpret spectra in general. Part 3 will cover an example of how we apply our Naive Bayesian classifier approach to this data, and Part 4 will end with a discussion and some short conclusions on where we go next. So let us proceed on. As a general in most Jupyter Notebooks, the first thing we're going to do is import our libraries. If you've not used Jupyter Notebooks before, then I suggest that you look at some of the earlier lessons that we've got in this batch of training. Whenever I've run a piece of code, there's a small number next to it. I will use these numbers to refer to the boxes as I go. So I will call this box 2. And in box 2 we import all the standard libraries that we have that we will use as part of this training code. This includes things such as importing data, libraries to control plotting such as matplotlib, and most importantly our sklearn.naivebayes class, from which we are importing the Gaussian NB method, which we will use to build our model. Following that, we are importing a few defined functions that we ourselves have written to help with this code and this Jupyter Notebook, just to remove some of the complexity from this demonstration. But you can actually find this ocean support functions _4f.py module in the same folder as this notebook if you want to take a little bit of a look under the hood. So let's move on to our first section, introducing ocean color and AltG marine data. Now I'm not going to read through all of this preamble because there's quite a lot of information here, but to give you an idea of why we are looking at this data. The ocean color signal is made up of light reflected from different components in the ocean waters. Innately we have an understanding of this, where we see water as clear, it generally tends to be quite blue. Where we see water as brown, it tends to be embedded with or contaminated with or mixed in with sediments. And when we see it as green, it tends to indicate that there might be plant life present, i.e. phytoplankton. However, our eyes are not very good at discerning the differences that each one of these individual components might contribute to the colour of the water. AltG, on the other hand, is very, very good at this process, whereas our eyes can divide this colours into red, green and blue, ALTRI has 21 individual wave bands that it can use to extract quantitative information at a given wavelength. The ALTRI land colour instrument is aboard Sentinel-3 and it samples most of the global oceans daily at 300 metre resolutions. It is our ocean workhorse, if you like, is the ocean workhorse of the Copernicus fleet. And these has very, very narrow bands, making it particularly suitable for work over the oceans. Now, the ALTRI data is distributed by EUMETSAT at 2 levels for the marine environment. There's a level 1 and a level 2 product. The level 1 product is a top of atmosphere product, which means it's not yet been corrected for the atmosphere. The level 2 product has the atmosphere correction applied, which means what we're seeing is what we see reflected from the surface of the ocean. When we talk about ocean colour, really what we're talking about is the combination of the intensities or the reflectances of all of these 21 bands combined together. And we have certain recipes, which we'll discuss later on, of how we take these 21 bands and combine them into something that's analogous to what our eyes would see. You can find much more information on all of this in the ALTRI Marine User Handbook. So let's move on and set up our problem. The first thing we'll do is a small bit of preparation to run our code. In box 4, all we're doing is defining how we control our images in terms of plotting and in terms of where we overlay masks such as land poly that we get from natural earth. For a level 1 image we still have the land in there, but for a level 2 image the land is flagged out and we might want to give ourselves some polygon that gives us an idea or a reference point of where our ocean corresponds to coastal features. We're going to look at these spectra over some defined regions, and box 5 defines these regions. Spectral box extent constructs 3 boxes, and spectral box col, or spectral plot cols, gives us the colours of those boxes. These boxes are the areas we will extract and average our spectra over, and the colours just tell us the colours that will be on the plot. The colours do not tell us anything about the colour of the waters in that box. And the last variable we'll set is this band max equal to 760. This means that we're going to constrain the training of our model to a maximum wavelength of 760 nanometers. Our training data actually goes significantly further than this, but we're actually going to constrain it to 760. Okay, so let's proceed on. The next thing we're going to do is load in our example data and take a look at it, just to familiarize ourselves with ALTRI level 1 and level 2 products. The data path variable we define in box 6 just points towards where the safe format .sen3 directories are for each of the ALTRI products, and you can see we have these S3A OL1 underscore EFR level 1 product and this S3A underscore OL underscore 2 underscore WFR level 2 product. These are the standard formats, the standard names that we would expect ALTRI data such as that on Wekio to be delivered in. Now, to get an overall picture of how these regions we're looking at seem from level 1, top of atmosphere, and level 2, we are going to extract red, green and blue channels from these level 1 and level 2 products. Now, this is not quite so straightforward with ALTRI. We could just map the nominal red, green and blue wavelengths from the satellite to red, green and blue, but this doesn't necessarily give us a great idea of what our eyes would see, as our eyes actually tend to integrate data over quite a wide spectrum of wavelengths. And we have a special recipe we called tri-stimulus that we can use to apply to ALTRI data to allow us to map the 21 bands that we're interested in to a RGB profile that matches our eyes. In fact, we don't use all 21, we only use 12, but that's what box 7 does. Box 7 will read in all of our reflectance data and give us a green, a blue and a red channel to map, and our longitudes and latitudes. As I said before, if you wish to look a little bit more about what's under the hood, you can look at this ALTRI support functions Python module. Box 8 just rearranges these red, green and blues to an image array so we can plot it. Nothing amazing happening here. And finally, we'll plot both of these images with the CartoPy library. So this next box, which would be box 9, will give us these 2 nice plots of what our region looks like, and we can compare them. So on the left hand side we have a level 1 image, this is top of atmosphere, and on the right hand side we have our level 2 image, this is as if the atmosphere were removed or corrected for. And we can see notable differences. Firstly, we'll note that the image on the left is significantly bluer. This is the effect of the atmosphere, it tends to make all of our features look significantly more blue. And the picture on the right is significantly more vibrant because once we've removed that atmospheric component, we can start to look a little bit more at the actual colours of the water. And we'll notice from our three boxes, our orange, our purple and our cyan box, that in both cases, but much more clear on the right hand side, we can see there's all kinds of interesting complexity happening in the water. For instance, in the orange and the purple boxes, we can see the influence of coastal sediment or river outflow from potentially the Thames estuary. And in the cyan box up towards Skagerrak, we have much clearer, potentially clearer water. We'll look at that in a bit more detail in a minute, associated with this zone over here. And everything I've just said really is reflected in the text you'll see below, so I'm not going to discuss that too much. So next, let's look at interpreting our ULTRI spectra. So what we're going to do is we're going to take our ULTRI bands and we're going to take our regions of interest and we're going to extract our bands across those regions. So box 10 will calculate, or will extract rather, from the Sentinel-3 metadata file, this xfdu manifest.xml, the bands themselves, their wavelengths and their bandwidths. And when I say bands themselves, I really mean their names. Once we've got that information, then we're going to read in the actual reflectances associated with those bands from the various netcdf files inside the safe format that correspond with each one of these reflectances. So box 11 will loop through all of the various channels. They're called OA, O1, OA, O2, etc., extracting over our region of interest the given information and taking a spatial average. So we should get three little processing box indications saying that all three boxes have been read in. And now we're going to plot these spectra. So box 12 will just take this information and plot it, and so we can see what the reflectances are and over what wavelengths. So from figure 2 we can see that we have notable differences between our three boxes, and the colours of the traces on this plot match to the colours of the boxes on the level 1, level 2 images in figure 1. We can see that the orange and the purple box have significantly higher reflectances in the 400 to 700 nanometer range. This is usually indicative of higher scattering, so that there might be sediment present in these waters, whereas the cyan coloured box is much more absorbent. There is much less reflectance in the lower part of the wavelength spectrum between 400 and 700 nanometers, indicating much less scattering, probably much less sediment present. However, we can also see this interesting little feature up around 700 nanometers, this little bump if you like, in all three cases, which indicates that there might actually be chlorophyll or algal activity present in all three of these spectral samples. However, these are challenging for us to interpret. There's a lot of information encoded in these spectra, and our eyes certainly are not very good at extracting all these individual pieces of information to use in a qualitative or quantitative way. Also, there are somewhere in the region of 1 million of these spectra over every given ULTRI scene, so it's not something we can approach by eye in terms of classifying what might actually be in the water. The third thing is, and we'll come to this later on, that we don't actually have a great idea in some cases of the specific constituents that contribute to this individual trace. This is something we call the spectral ambiguity problem, and we'll discuss it at the very end of this notebook. There are lots of different ways for us to construct a spectrum of this type in all three cases that you see in figure 2. So there are some challenges here, so let's see what we can do with machine learning to address some of this complexity. So what we're going to do with our naive Bayesian classifier is classify our waters into two types, and we're going to call these types case 1 and case 2. Case 1 waters are waters that are a little less complex, and most of the spectral signal that we see is associated with a concentration change in algal properties, so a concentration change of chlorophyll. The case 2 component is where we have lots of mitigating factors that might mess all that up. We have influence of sediment, we have influence of CDOM, we still have algae, but things get a lot more complicated. So we're going to try and divide these into case 1 and case 2, which we're kind of roughly going to call relatively simple and case 2 complex. So we are going to classify things as either simple or complex. Now, in order to do this, we need to build a model, and in order to build a model, we need training data. Now, in order to train our data, we're going to use the output of a radiative transfer model that includes an IOP model which defines how our model will create spectra from known input properties. So the model we're going to use is based on the data set from Eversking et al. 2014. The way it works is we define what's in the water, and then the model will tell us what our spectra, our RRS, our remote sensing spectra, our remote sensing reflectance spectra should look like based on those in-water properties. So this is our known quantity we're going to use to train the model. All this information is contained within this model spectral reflectance data netcdf file. This is our training data, and we're going to read in a few variables from that. We'll read in the wavelengths for this model, we'll read in the RRS, so our modeled remote sensing reflectance, and then we're going to read in our flags or our labels. So we have two, and they're actually mutually exclusive, so we only really need one, but we'll read in them both. One is called EcoLNLG. This is our flag for where we have low non-algal, low CDOM waters. So this is where algal populations dominate the reflectance spectra. Then we have one called EcoHNHG. This is our flag for high non-algal, high CDOM waters. So irrespective of the chlorophyll concentration, we will have lots of scattering from sediment, and we'll have lots of influence of dissolved organic material as well. So let's plot these data and see what they look like. And box 14 will plot figure 3. Now figure 3 shows us that in blue we have our LNLG, so blue is where we have chlorophyll concentrations only, and red is where we have the influence of sediment and this CDOM material. And you'll notice first and foremost that the influence of the scattering from the non-algal particles gives us a very, very wide spread in terms of the spectral shape, whereas the blue traces corresponding to the LNLG scenario are significantly more absorbent and have much lower reflectance. On this plot, I would also point out that our training data is in 5 nanometer increments, whereas our ALCHI data has only these 20 or so bands, and the ones we're going to use are shown in these grey stripes. So we're going to use 2, 3, 4, 5, I think 12 bands in all when we construct our training data set. So let's go ahead and train our model. So the first thing we're going to do is going to read in this training data we've got, and then we're going to limit it to our 760 nanometer band max we defined earlier, and we're only going to read in the wavelength bands that correspond to the ALCHI bands. So everything else on that 5 nanometer incremented scale we're not going to use, we're only going to use the ones corresponding to ALCHI. We're then going to split this data into 2 halves. We're going to retain 2/3 for training our model, and then we're going to retain 1/3 for testing our model once we have trained it. So this little test indices variable here is set to true wherever we've taken everything 3. This is every third index will be a test index. Constructing our model, our naive Bayesian classifier, is incredibly simple using scikit-learn. We imported this Gaussian NB model from the top in our first box, and all we have to do is instantiate it by saying GNB, variable name, is equal to Gaussian NB, no arguments, and our model is now ready for us to begin to configure. In order to construct our model, we use this gnb.fit method, and we're going to call it using data array, where our test indices is false, so our training indices nominally is true. And then we will call it with the reflectance data, and then we'll recall it with the HNHG indicator also being false. So this is our data and these are our labels. So we have all of our reflectances and all of our 1s and 0s. And that is it. That is the scikit model constructed for this training data set. But let's just have a quick look at how this scores with our remaining testing data. If we run class model.score with the opposite, so test indices is true, our final third of the data and our final third of the labels, we get a score of about 0.73. This is a reasonable fit for our model. It's not so low as it will be functionally useless to us, and it's not so high as we start to be suspicious that our model was overtrained on our training data and therefore possibly not fit for purpose outside of the confines of this data set. Moving on, we'll now apply this model to our image. So all we're going to do is we're going to read in, as we did before in box 19, all of the reflectance data associated with each one of our ALCHI channels. So when we run this box, it will tell us it's reading in everything from 400 to 753 nanometers. In this case, we're not reading it over a spectral box because we're going to apply this to the whole image. And we use this data in box 20 to actually construct our final output classified array of where we think we match to case 1, simple, case 2, complex waters. So box 20 will do all of this. We set up an output array. We flag this array to get rid of anywhere where we're not happy with the quality of the data, i.e. it's flagged due to cloud and therefore we wish to exclude it. And then we classify it based on our model using this class model.predict method. And we send in this image array QC, which is just our reflectance data but flagged. And then we do a little bit of refitting to the original size of our array, just because we send this as a one-dimensional image, but we actually want it to be in 2 dimensions for when we plot it, which is what we're going to do in the next box, in box 21. So when we run box 21, we're plotting, as before, our level 2 AltG RGB image. Remember, this is atmospherically corrected with the same boxes as before. And on the right-hand side, we have our classified scikit-learn output, where case 1 is blue and case 2 is red, using our trained model. And you can see some interesting components happening here. When we look at the picture on the left-hand side, we can see that both the orange and the purple boxes tended to be more yellowish, brownish. And when we compare that to what we see on the right, we can see that those actually correspond to quite a lot of coverage of case 2 complex waters. So this kind of fits with our preliminary a priori understanding of what we thought was happening here. And in the cyan box case, we have a situation where the box was a little bit more ambiguous maybe, but it's probably not quite as brown as the other two before we began this exercise. And this scikit-learn, the model that we've made, has told us that this is probably dominated by case 1 waters. It's predominantly blue. It's probably algal populations here that are controlling our signal. So just using this simple classifier, we've been able to derive some relatively sensible looking spatial patterns of where we might be dealing with quite a lot of optical complexity in our waters and where we might be dealing with something a little bit more simple to deal with. So as we move on to the conclusions of this exercise, before we actually sign off, we're just going to ask ourselves a question: is this actually useful? Is this sort of water typing something that can give us information? Case 1, case 2 water typing is a basic form of classification. It has a long history in the colour and the science of ocean colour, and you can read a lot more about it in Mobley et al. 2003. It is generally a first pass approach for us to be able to gather preliminary information about whether or how we treat these waters when we're looking to perform something maybe a little bit more complex. But it does have its limitations, this approach. Number one, case 2, the whole idea is a construction that helps us, but in reality, there's a whole spectrum of waters, a whole grading of waters between case 1 and case 2. There are many in-between water types. There's not just a binary classifier. Secondarily, the scikit-learn Bayesian approach relies on the classes being independent from each other. And this may not be the case where we have significant mixing, such as the tidal regions of the North Sea, where things are quite near each other. We might have a little bit of overlap. Thirdly, we're only saying, yes, this is complex. No, this is not complex. We're not, in this case, deriving anything about the concentration of the in-water products, which is a problem we'll come to in following Jupyter notebooks. So in short, it's a simple classification that can be conceptually useful, can be beneficial to us in terms of how we deal with the complexity of ocean waters, especially near the coasts, but we do need more accurate approaches if we want to attempt anything more complicated, such as a more refined classification, or to actually retrieve in-water concentrations of, say, chlorophyll or algae or sediment. So in conclusion, for decision-making activities about our coastal oceans, we often need information on water quality. Satellites can provide us this information, but often this information is really, really complex. The mix of effects from different in-water constituents can give us a very complicated picture. However, using machine learning approaches such as scikit-learn, we're better able to classify these waters to know when we need what tools and when we can apply them, giving us much better information, allow us to further the science to go a little bit further in terms of deciding how we best manage our coastal zones.